at least maybe let's provide some Zoom instructions, and then
Okay.
I'll go.
Yeah, let's start with that. All right, so welcome everyone to our second K-CART research seminar for the semester. So just a few housekeeping items to start out. So we do ask that you all remain muted during the presentation. If you all do have questions, you can, for Dr. Veatch today, you can ask those in two different ways. You can either send them via the chat, and you can do that at any point during the talk, and then Matt or I will pose them to Dr. Veatch at the end of the talk. Or if you wait till the end, you can also unmute yourself at that point and ask her directly. And then our next talk is going to be with Dr. Tomey on October 22nd. So that's 3 weeks from now. And I'll be sending out initial invitations for that in about a week. And I also want to mention that you all can find the complete schedule for our seminar for this semester on the K-CART website under the events tab.
So hi everyone, this is Matt Mosconi. I'll be really quick, and then we'll let Shannon introduce our speaker for today. But very briefly, as Shannon referenced, the seminar schedule for the remainder of the semester is posted on the K-CART website, so I'd encourage you to check that out. But briefly, we do have, after today, 3 more talks scheduled for the semester, including Lauren Tomey, who will talk about physical activity and exercise in individuals with IDDs. Shannon Kelly, who you see in front of you, is a graduate student in the clinical child psychology program, and she'll be talking about her work on cognitive and sensory motor issues in autism and their familiality. And then Nicole Zieber is going to discuss her work on, I believe, infant emotion detection and attention. So a diverse set of talks upcoming for the semester. And then also, I will say that if others who are here, or if you have colleagues who are not here and you want to volunteer them, we are looking for volunteers for speakers for next semester. So feel free to throw your colleagues under the bus, or to contact Shannon or me directly if you would like to be included as a speaker in this series. So thanks for that. All right, Shannon.
All right, so I'll introduce our speaker today. So today our speaker is going to be Dr. Olivia Beach. Dr. Beach got her bachelor's degree here at KU, and then went on to get her master's in molecular biosciences and bioengineering from the University of Hawaii, Manoa, and then got her PhD in human genetics from Vanderbilt University. She then completed postdoctoral fellowships in neurology at Vanderbilt, and then in medicine at the University of Pennsylvania. She just recently joined the KU Medical Center faculty just earlier this year, and she, after she left her position as a research associate at the University of Pennsylvania, and she's already becoming very involved in the autism and IDD research community here at KU and KUMC. And she is going to be acting as co-director with Matt Mosconi of the Clinical Translational Core of our KIDRC. Her research interests include using computational genetics, bioinformatics, and in vitro studies to help promote the use of genetics in precision medicine. She's also very interested in characterizing pleiotropic genetic effects that influence the expression of comorbidities, such as sleep disorders in neurodevelopmental and neuropsychiatric conditions. And so today, Dr. Veatch is going to be talking to us about deciphering clinically meaningful genetic effects influencing autism spectrum disorder. So you can take it away.
Okay. Okay, so first of all, I just want to say thank you for inviting me to get to do this. And I was just saying with Matt and Shannon how I would have loved to have been able to actually meet everyone in person in Lawrence. Hopefully that will be a possibility in the near future. So I'm looking forward to that. And I just want to talk a little bit about some of the work that I've done during my postdoc and what I'm currently doing, and kind of hopefully give you a snapshot of what I do and where I'm looking to go as a new independent researcher here at KU Med, and keeping in mind that anybody who's interested in talking to me after this, please feel free to email. Love collaborating. I especially love collaborating with clinicians because I am a geneticist. Most of my training is basic research and computational genomics, and so it's really informative to be able to interact with people who are, at the forefront doing the sort of work that I'm hoping to inform. So I'm gonna start by talking a little bit about this lofty goal of precision medicine. So what we tend to do right now, and actually what was primarily done in the past, is this traditional approach to medicine, which you test a drug or a treatment strategy in a small group of people, and then everyone gets that standard care once it's been approved. So in this case, you're going to have some people who respond appropriately to the pharmaceutical compound, and primarily those are the people where the compound was tested. And then you'll have a subset of people who have adverse events and others where it will be completely ineffective. It's now gotten to a point where we're doing more of a stratified approach to medicine. So you're characterizing people based on common traits. So men, women, children, white, Black, Asian American, and then you test treatments in each of those groups and you'll modify the treatment accordingly. And the goal of that is that then we would have an approach that is effective in the majority of individuals where you're using the treatment and we're minimizing these adverse events. But the real goal that we have for future work is to do this more precisely. So we're going to incorporate genetic information, environmental risk factors, lifestyle risk factors into how we test our treatments. And that way everyone maintains individuality. All treatments are tailored to the individual. And then the goal is then that this will be effective in every single patient with no adverse events. And I say that's a lofty goal because this is something that I think is not going to be in the near future. This is something that's going to be quite some time into the future, especially incorporating genes, which is what the goal of my work is. So to incorporate variability in genes, I like to kind of group how genetic associations are done and how the genomic data is generated into these classifications for precision medicine. So traditional medicine would be what we would call a genome-wide association study, where you just take one group of people who has no evidence of disease, another group of people who has evidence of disease, you lump them all together, and then you look to see what are the genetic variants that are more common in the people with disease. Stratified medicine is now the approaches that are being used here are more along the lines of polygenic risk scores, where we're building cumulative risk most of it's based on results from GWAS, and trying to understand the individual's risk based on the overall evidence that they have genetic variants that are associated with their disorder. And then precision medicine is where we're really going to be starting to use techniques like whole exome sequencing and whole genome sequencing. So there are a number of barriers that are impeding us incorporating genetics into the clinic today. One of those is that there are thousands of variants implicated in complex disease. So this is a screenshot of the current GWAS catalog. You'll notice that every single one of these is a chromosome. These ideograms are chromosome. Every single mark along this chromosome is a single nucleotide polymorphism or a copy number variant, where there's a genome-wide significant associated trait. And then each of those little colored circles along each of those is a different trait that's associated with that variant. And they're all different types of very clinically distinct conditions. All different levels of underlying biology can be associated with the same genomic variation. So there's also this genetic heterogeneity. So you're going to have different variants that are influencing the same trait. Most of these variants that we're identifying, even a number of these de novo variants that are a hot topic in autism genetics, don't have really large effects. And a lot of these effects seem to be cumulative, which makes some sense because if one change could disrupt us entirely, we probably wouldn't have evolved to this point as humans. It's also difficult to identify effector genes. And this is a point I want to make because this is kind of some more recent data that's coming out of metabolic syndrome studies. So the good example is the FTO story. So FTO, there's a variant in the intron of that gene that is very well characterized for being associated with obesity. They started doing a lot of functional studies for FTO, assuming that the variant in the intron of FTO that's associated with obesity was actually tagging FTO. And what they ended up realizing when they started doing chromatin interaction and knockout studies in mice is that the variant that's in the intron of FTO is actually sitting in an enhancer region that's regulating another gene that's thousands of base pairs away. So just assigning a genome-wide significant hit to the gene that's encoded in the region that's nearest that hit we're starting to find out that that's really not the most appropriate way to define these functional relationships. Another thing that impedes us being able to use genetics data in the clinic is this phenotypic heterogeneity is difficult to model. And so one of the goals of some of my work is to try to elucidate pleiotropic effects. So the pleiotropic effects that I'm talking about is truly when dysfunction in the product of one gene contributes to expression of two clinically distinct traits. And so trying to find that information, you can imagine, would be useful because if then you can say, for example, in autism, if you have someone who has autism and they also have a sleep problem, if there's a common underlying genetic mechanism that you can then target to treat the sleep problem in that individual, that would actually be potentially more effective than trying a bunch of different compounds without any focus. So I just say that precision medicine is really well incorporated into cancer. And I would say that cancer treatment is probably the poster child for incorporating genomics into precision medicine. And so if cancer is the poster child, autism is like the black sheep of the family. So we're talking about an incredibly heterogeneous condition. The prevalence is very high, as all of you know. The heritability that's been estimated in autism really suggests that it's common inherited genetic variation that's influencing expression of most cases of autism. The family-based and linkage and GWAS have supported this. And then one thing that I like to point out, and I'm sure all of you know this, but I think this is something important, is that the way we define autism really is along dysfunction along a spectrum of behaviors that typical people express. So everyone expresses social behaviors, and honestly, most people express some level of restricted and repetitive interests. It's just at a point where it's atypical that you then have a diagnosis. And so then I also like to add this to the mix after saying that, is that there's a really interesting study that was published in 2016 in Nature Genetics, which looked at the prevalence of risk variants for autism in the general population. So people who have no evidence for autism, these variants are all over the board. Most people carry a vast number of them. And then getting more at this de novo versus common inherited variants. It's also particularly interesting to note that when individuals have de novo variants, They may not have autism, but they have, when the burden of de novo variants increases in a typically developing individual, they start to express traits that are more indicative of how we define autism. And so just to add to the complexity, so this is our landscape that we're working with. So I would love for someone to just come in here and show me along this ring plot where I can find genomic information that's going to help a clinician understand better how to treat his or her patient with autism. And so that's one of the goals of my work, is to try to focus in on this. So I'll leave the identifying novel variants to other groups. And then what I'll do is take that information and try to find creative ways to pinpoint the most clinically relevant information. So one way I do that is to focus on comorbidities. So this is, I love this figure, and it's really good at explaining not just the genetic heterogeneity, but the phenotypic heterogeneity that's seen in autism. And two things, they've missed one really important comorbidity here, and that is insomnia. And so insomnia is actually reported in as many as 80% of kids with autism, but it's really, the lower bound is almost half. So it's a very common comorbidity. It's actually one of the most common comorbidities that has been reported. So this actually indicates that there may be some underlying disruption in how sleep is regulated. And so I'll just briefly touch on this, but I'm happy to talk more about it if you're interested. But we all have, every cell in our body has a clock, which is a genetically encoded oscillator that is basically a timekeeper, and it helps us respond to our environment. And so what this clock does is helps us know when to fall asleep and when to wake up in response to light-dark cycles. It also, in response to those light-dark cycles as well as temperature cycles, it tells us when to release hormones, when our temperature should go up and down. And so this is like a very dynamic, constantly oscillating system. And what has been observed is that numerous individuals with autism suffer from sleep problems, body temperature regulation issues, and hormonal variations, all indicative that there may be something disrupted in this underlying clock. And I want to point out this study because it actually, there's some question as to causality here. So does the sleep problem cause more severe autism? Does the autism cause the sleep problem? Is it a vicious cycle that's occurring? And in this case, these individuals pointed out that from most of the literature, it looks like these circadian disruptions are preceding the diagnosis of autism. So they're starting to observe these in the first year of life, when the autism diagnosis is not coming until at least year 2. There's also specifically evidence for involvement in the melatonergic system. So now we're starting to pinpoint a mechanism, and melatonin is required for our circadian regulation of sleep. So this is the first study that really looked at a relationship between genes that are encoding enzymes in melatonin biosynthesis pathways and a relationship with autism. And so what they saw was that there were actually polymorphisms in the promoter element of the gene that encodes the rate limiting step in biosynthesis of melatonin. And that when you had a haplotype that was comprised of 2 different SNPs in that promoter element, that was associated that was preferentially transmitted from parents to children with autism compared to children who did not have autism. So that's what the transmission disequilibrium test there, the TDT. Not only that, but these polymorphisms in the promoter element appeared to sit in the locations where transcription factors should be binding. So they thought that maybe they were disrupting transcription factor binding. And they did see that when you had the autism-associated genotypes at these 2 SNPs, that you had lower levels of this enzyme being produced. They then tied that into the actual activity of the enzyme. So once it's encoded, So now we're getting at the protein level. And then the ultimate product, which is the most of interest, is the melatonin. And so what they did see was that this was kind of interesting. They saw that individuals with the autism-associated genotype had a correlation with melatonin production if they were diagnosed with autism. So that was distinct from people who, these are actually the common variants. So these are the most common alleles at these SNPs. So a lot of us probably have these. But what they were showing is actually if you have them and you don't have autism, it's not correlated with your melatonin production, which is kind of interesting. They then replicated this in some larger studies, which is important. And then, so I did some work trying to decipher some of this information. And so we actually wanted to focus on individuals who had autism and comorbid insomnia, because a lot of the studies that tried to replicate this finding didn't see a replication. So they did, the same group was able to replicate, other groups weren't able to replicate the association. And the common theme was that nobody was focusing on sleep problems. So nobody was focusing on identifying this subset of individuals who have autism and comorbid insomnia. So that's what we did. These are clinically defined with sleep onset insomnia. And we did see that the genotypes related to the lower levels of this enzyme were more frequent in kids with autism and sleep onset insomnia. The interesting thing was that these individuals did respond to a low dose of supplemental melatonin. So their sleep onset insomnia improved. Their symptoms of the autism that were exacerbated potentially by this sleep onset insomnia also improved upon treatment with melatonin. However, these individuals had a normal dim light melatonin onset, and they also had normal endogenous levels of melatonin. So what I then did was looked at the enzyme that metabolizes melatonin, so it's downstream, and so this is what's metabolizing melatonin in the liver. There are slow metabolizing alleles that have been well documented in this enzyme, and so I looked at those slow metabolizing alleles specifically and saw that not only did individuals have genotypes that were associated with lower levels of the enzyme that produces melatonin, they also had genotypes that were associated with slow metabolism of the melatonin. And so that was actually something interesting. So this is just showing that haplotype plot across the two genes, and just this strong correlation between the SNP in CYP1A2 and the SNPs in ASMT that show this relationship. And so the thought was that maybe what's happening is that these individuals are producing less melatonin, but they're also metabolizing it slower, so it maintains a normal level endogenously. And so then that begs the question, then what's the melatonin doing? It's not treating a low endogenous level of melatonin. What is it treating? So that's some work that I'm still doing, and that's a question that's still up in the air. There's actually a recent study that came out in the mouse. It's a mouse model of catnip2, where they actually showed that treating a mouse who has that gene knocked out with melatonin improved repetitive behaviors in the animal. So it's just interesting to think about this concept of if sleep might be this modifiable risk factor. If you can focus on treating the sleep problem, you may actually improve a number of other behaviors. So then I tried to validate this. We didn't see validation in a larger cohort. And what I would like to point out is I think that was actually because we were dealing with issues of reliability. So the first data set where we saw an association was a very well-defined data set. They'd gone through a number of tests. They had ruled out all these other comorbidities, all medication use, et cetera, and et cetera. The other data set where I tried to validate and didn't see anything was solely based on parent report. So I said, okay, well, what can we get from parent report? How reliable is it? So I used the data set that we had where we actually had actigraphy, which is an objective measure of sleep behavior, and parent report, and so I could do a comparison. And what we saw that when parents report problems with sleep, they do not predict differences when we measure that with actigraphy. And that the only thing that was correlated from parent report and what we could measure with our actigraphy was when I actually took the parents' report of when they said their child went to bed and when they said their child woke up and calculated a sleep duration based on that. So then I was able to take that and then go into a big data source. So now I basically went into the Simon Simplex collection, which is one of the largest collections of simplex families, and I took the only sleep data that they collected of all the sleep data, one question, which was how many hours per night does your child sleep? Because that was the only thing I'm truly trusting is reliable across a large population like this. And what we were able to see was that shorter sleep duration reported by the parents is specifically associated with more social impairment, and that was primarily an issue with developing peer relationships. And I'll also note that that's seen in typically developing individuals. And then also this increase in compulsive non-functional rituals, which would be related to some level of repetitive behaviors. Now, I also saw that more maladaptive behaviors were being reported in individuals who had shorter sleep, and also that a number of comorbidities were more commonly diagnosed in individuals who had shorter sleep. So there may be something going on here where the quality of sleep for the individual early in life, especially an individual who has a neurodevelopmental disorder, may impact their function long term. So if the sleep problem is causing more severe symptoms of the autism, what's the underlying mechanism there? And so I think that there is the potential that this relates to synaptic balance. So I'm just showing what I'm sure a number of you have seen before, but this is the evidence that there's decreased pruning in individuals with autism in the temporal lobe. There's also some evidence to the counter of that, but it's actually just atypical pruning of synapses. So sleep, one of the reasons we think we sleep is because while we sleep, pruning is happening. So appropriate pruning is going on as we sleep. So in different stages of sleep, we're pruning synapses in certain regions of the brain, and in other stages, we seem to be building our synapses. And so sleep is this time during our lives, which is really critical to controlling the appropriate plasticity of synapse development. So then this is a report that came out focusing on genes that are associated with autism and trying to categorize them into biological networks. And what they saw was that there's a network of genes that are strongly associated with autism that are all involved in synaptic function and synaptic plasticity. They did see that there were two different groups, so that individuals who had issues with synaptic genes, de novo variants primarily, didn't have any evidence of dysfunction in this other process, which was related to chromatin remodeling and the vice versa. But they didn't actually see any symptom differences when they compared those two groups, even though they were genetically distinct. I'll point out they didn't evaluate sleep because I actually asked Dr. Sanders when I saw him give a talk. And they also didn't consider all variants. They really focused in on what they thought were more likely to be high effect variants, which are de novo or really rare inherited copy number variants. And so if there's some cumulative level of risk, incorporating common inherited variation may actually be useful to better understanding a relationship between synaptic gene dysfunction and expression of things like sleep problems. And so again, this is the largest GWAS that was ever done when it was published. This is the first GWAS that came out of the UK Biobank, and they focused on insomnia because everyone in the UK Biobank had some level of reports for their sleep quality. And what they saw was that a number of synaptic genes, many of which overlap with neuropsychiatric conditions, are associated with insomnia in this large data set. And this is just kind of honing in on these biological processes. So if you take all of these genes with variants that are associated with insomnia and you group them together, what are we seeing? We're seeing that it's synaptic plasticity that seems to be disrupted in individuals with insomnia. So I'm actually trying to tease that apart. I'm not going to talk more about that today, but that's one of the projects that I'm doing right now. Other things that I would love to have done to support sleep genetics and autism is to find better ways to identify sleep issues. So coming up with more reliable traits, whether that be objective measures, or informing parents, because parents did improve in their reporting of sleep issues when they were just told what's appropriate sleep hygiene. And then I'll just note that as to add to complexity, there's also evidence that genes that are controlling sleep during development are distinct from genes that are controlling sleep in the adult organism. So we're talking about kids. We want to start treating sleep problems early on. We may have to focus on a different subset of genes or different ways of elucidating what genes are controlling sleep during development of the organism. Okay, so ultimately, these are the sorts of studies that are being done. All of this information is out there. What's another barrier that impedes us getting this information into the clinic? Well, that's primarily related to the people who are actually going to have to incorporate that information, and those are clinicians. So clinicians do not have time to go through and comb the literature and try to decipher all these like little effects and all of these biological studies and all of these large de novo variant association studies. So one of the goals of my, the grant that I'm working on now, so this is a KO1 award I got through the National Library of Medicine. And this I brought with me here. I'm in my last year of that. So the goal is to develop a tool that can pinpoint and summarize the genetic information that's relevant to informing treatment, and to produce that, present in a manner that's conducive to translation. So I'll tell you how I'm going to do that, or how I am doing that. So all of these issues that we have, I'm only addressing two. One is the immediacy gap, so how to get that information that's all on the internet compiled together, organized, and put into a common place so that it's easy to access. And then the processing gap, which is how to take that information and put it into a format that's actually going to be useful to a clinician. And in this case, I define clinically actionable as something that's a gene that's functionally relevant, that has evidence indicating there may be some underlying syndromic condition, or that there's a variant that may influence response to a drug. And so these are information that, regardless of how strongly associated the gene is with autism, if there's a chance that this individual with autism may have a variation in this gene, and it's going to influence how that patient responds to treatment, or it may actually indicate that that patient has an underlying syndromic condition that you need to be focused on treating. That's the sort of information that we can get rapidly into the clinical environment. And the goal is to support these decisions. So I'm not making these decisions for the clinician. I'm just simply presenting them with all the information so that they can go in informed to determine whether or not they want to do genetic testing on a patient. Okay, so basically how I do this, so as was said, I do a lot of computational and bioinformatics work. So I do a lot of writing of code. So I start with a resource called DisGeneNet. This is a fabulous resource if you don't know about it. It basically compiles information from all over the internet. So these are all of the sources of information that is pulled together to say that there's evidence that a gene is associated with a disease. So you'll see that there's a number of different diseases, there's a number of different genes, and you get all the evidence compiled in one place, and it does that through natural language processing and text mining. So I basically access this programmatically, and then I dump it into my favorite software, which is R. And then I start a pipeline. So this is a almost fully automated pipeline. So basically the first thing I do is look to see how many of the genes that are associated with autism across all of these resources have evidence of being expressed in the brain. And that's a pretty straightforward approach. So one of the things I wanted to do with this approach, though, was to make sure at every step in my pipeline where I'm trying to annotate genes, that that's actually relevant to autism. And so to do that, I also took 1,000 subsets of genes randomly pulled from human protein coding genes that have been defined of the same size of my autism gene set. And then I ran those all through my pipeline as well to see how often my autism genes are showing up, more of the autism genes are showing up with that trait. So in this case, what you're seeing is a histogram of all the random gene sets. So you see a pretty much normal distribution. When you pull randomly, you'll see that there's a normal distribution of genes expressed in typically developing human brain. And the autism genes, there's exponentially more. So that's a significant difference. The other thing that's interesting is so keeping in mind this is typical brain, but we're talking about autism genes. So there's more autism genes expressed in the brain, and it actually seems to be driven by overexpression of autism genes in the pituitary gland. So I'd love to hear all of your ideas for why that might be. I have some of my own, but in a typical person who is arguably non-diseased and doesn't have a neurodevelopmental disorder, genes that are associated with autism are expressed at higher levels than random genes in the pituitary gland. So then what I did was focus in on overrepresented processes, so trying to hone in on genes that are all functioning in processes that might be relevant. So they're expressed in the brain and they function in a process we care about. I did that in mice and humans because then I took data from knockout mice. And so I looked and said, okay, so now we have overrepresented processes for autism genes like synaptic plasticity. What are the corresponding traits that are looked at in the knockout mouse phenotyping project? And so that actually is a large consortium, it's international, and the goal is to run a phenotyping pipeline that's non-focused. They test a lot of different traits on knockouts for every single protein coding gene in the mouse. That's the eventual goal. That's not done yet, but they're working on it. And so those data are actually programmatically accessible, and so I can pull them and say, how many genes are associated with a trait in the mouse that's reflective of a process that's overrepresented for autism genes? And what we saw was that it's actually primarily these growth and growth traits. So it's actually abnormal postnatal growth, that there are more autism genes that are associated with that trait in knockout mice. There's 63 autism genes from that long list of 956 that I started with that's associated with a growth trait. These are the most common traits that we saw more autism genes associated with. So just to note, say for example, that there's 29 autism genes all associated with decreased lean body mass when you knock it out of the mouse. Okay, so then I wanted to really focus in on clinical translation. So here I'm using information about FDA approved drugs and molecules that are similar to FDA approved drugs, as well as pharmacogenomic variants, and that's all information that I can pull programmatically. And so I'm looking to see if any of the autism genes in my list encode a drug target or a molecule that's similar to an approved drug target. We did see that more autism genes are approved or similar to approved targets. Also, not really anymore are just we know the biology of, so that kind of helps to say that we didn't introduce too much bias by starting with a list of autism genes where we know they're well studied. And then also that there's fewer autism genes that we don't know anything about, which are these dark genes. And there's also more autism genes that contain pharmacogenomic variants, which is really important. Okay, so finally what I did was want to see if any of the autism genes in my list encode a protein that is evidence to directly interact with a protein that's encoded by genes that we know have pathogenic variants. So these are things that are recommended by the American College of Medical Genetics. There's 59 genes. These are recommended to be tested for. And we did see that more autism genes have evidence for direct interaction with these proteins. And so if you take every single step in the pipeline and you run the list through it, you end up prioritizing from 956 genes, 18. Of those 18 genes, 14 had a pathogenic variant reported, or a likely pathogenic reported variant. So I'll note that if you take the full list and you just look at how many of those from the full list of autism genes have likely pathogenic or pathogenic variants, it's about 50-50. So I'm actually doing a better job, or at least I'd like to think I am, of prioritizing genes that are more likely to be clinically actionable. And here I'm just showing you in yellow the autism genes, in blue are the American College of Medical Genetics genes. So you're seeing this network here of prioritized genes, and the ones that I'm highlighting with the green box are the ones with pathogenic variants. And I want to just focus in on one gene specifically because I don't have time to go through them all. But I want to focus on one that you would not have probably looked at if you hadn't gone through a pipeline like this. So the angiotensin converting enzyme was linked to autism through one study. There's one PubMed publication on this. It was a case control study of 120 patients, so incredibly small, and it was a candidate gene study. So these are not the things that most people are going to start functionally following up, and they're not the things that most people are going to think are clinically relevant. However, ACE is expressed in the pituitary gland. That's the highest level of expression for ACE in typical individuals. It's associated with abnormal embryo development in the mouse. And I'm just showing you what the mapped human process is. There are 19 approved compounds that target this gene. I think everyone knows ACE inhibitors. I think we probably all know someone who's on an ACE inhibitor. And to assume that a patient with autism is never going to be on an ACE inhibitor is kind of ridiculous. And the other thing I want to note is there's pharmacogenomic variants that have been reported in this gene. It's actually considered a very important pharmacogene. And specifically, there are variants in this gene that have been reported to influence how a patient responds to fluoxetine or sertraline, which are both approved to treat anxiety in individuals with autism. So hopefully I'm making the case that this information would be useful to a clinician to help them understand. So maybe I want to do a little bit of digging, make sure a patient doesn't have these variants before I put them on maybe one of these antidepressants, or before I put them on an ACE inhibitor. Just information that's useful and could improve how you're treating the patient. So ultimately the goal is, and I'm actually working with the biostats group here at KU Med to build this. So I have like the skeleton of the database put together, but so basically I'm pulling from all of these pipeline, or all of these resources to go into my pipeline. I run the pipeline. That information is getting stored in a database. So you don't just get information on prioritized genes, you're getting information on where every gene has an annotation in the database. And then the idea is that a mobile app will be developed. So that's where you're getting to more efficient translation, because if a clinician doesn't have time, really, what can we do? Maybe we can get them to use a mobile app when they're on the train, when they're walking to their car, you know, when they're out at dinner, and say, oh, I saw a patient today with autism. Wonder if I should do any genetic testing. Let's see if there's any information out there that might be useful to me if I were to test for it. And so they can run this little cache database. That the idea would be that there'd be user-specific profiles. So if you're an autism clinician, you're only going to get information about autism-relevant genes. And that hopefully that will be something that's useful to help improve how you're going to treat a patient. And that's kind of just the output that would look. You'd see these check boxes. So gene A is checked in every category, and you can click on a link and that'll take you back to the source material so you can read more about it. So basically, my research program really as a broad goal is to build these bridges between what we're doing in basic science research and computational genomics research and the clinic. And so hopefully I made the case that I do that with things like characterizing pleiotropic effects. I do a number of approaches for that. I also use the electronic health record a lot. And so Matt talked at the beginning, actually, I think it was Shannon who mentioned it, that I'm being involved with the CTC. And one of the goals is that we're going to be building resources that allow us here at KU Med to do research in the health record, where we're linking health-related information with things like biorepositories so we can do genetics using EHR data. And another thing is looking at genetically meaningful phenotypes. So I do a lot with machine learning and unsupervised approaches to try to decipher these really complex traits. Are there any symptom profiles that are distinct? And ultimately build these tools to help automate these prioritization approaches. And I'll just plug at the end. So I did do a lot of bench work initially in my career, and I haven't done so much lately, but I'm really interested in getting back into characterizing some of these pharmacogenomics variants, especially those that are being prioritized using in vitro methods, because the sooner we can start focusing in on this and deciphering these signals, the better for our patients. And I'd like to thank a number of people who've helped with the projects and throughout my career and my funding, and particularly all the individuals who participated in all of these projects, who helped generate these data, without which I could not do anything. So with that, I'll just end and take questions.
Excellent. Thanks so much, Olivia. It was a great talk. A lot of information and a lot to think about, I think, for those clinicians in the audience, as well as those of us who do more of the different clinical characterization of autism, as well as looking at different intermediate phenotypes, which you've pointed out the need to really identify more objective markers that may be linked to some of these different variants. So with that being said, though, let's open it up to questions. So if individuals want to either send questions through the chat or unmute themselves and hopefully turn on your video as well and ask Dr. Veatch directly.
Matt, I'll start. This is Steve.
Hey,
Can you hear me?
Steve.
Yep.
Good. So fascinating talk. The tools you're bringing to this are impressive. It's obviously a complicated world for sure. And so that brings the issue. I just want your thought on this. You know, the word you hear most of the time now, or at least I hear a lot of times, is the autisms. and I think it makes sense that people talk about it that way, because you have to carve this out somehow. The work that I work on and my colleagues here is Fragile X, which is really relevant to this. But it is clearly has differences that are related to being Fragile X, and that said, it's still. Even though it's probably farther along in terms of medication kinds of things and stuff like that in many respects, it's got a long ways to go. And there's a lot of things we don't understand still. So how are you thinking about the autism in terms of your own work? Are you trying to cover in a way, here's 5 different autisms if you run this kind of big data stuff, which is sort of, I think, what you're doing? Or are you sort of going, no, I'm not going to try to do that. I am interested in some of these issues, but I'm going to keep this to a certain group of folks who bring enough pieces of this together that we think we can make a sense of it.
So I'm glad you asked that. So actually the goal and the reason that I used a number of these resources, so you could probably pick on every resource I used. So for example, let's talk about GTEx data. Those are typically developing individuals. It's not that we're looking at what genes are differentially expressed in an autism brain versus a typical brain. We're looking at genes that are expressed in tissue that was collected from postmortem human samples with no evidence of disease. And so arguably it would be more relevant to take a pipeline like this and try to prioritize based on genes that are differentially expressed in autism brains. And maybe those are more interesting. But the goal of this was to build a pipeline that could be a backbone for expansion. And so those are the next steps. And that's why with this database, we're going to have different pipelines for different diseases. I am primarily focusing on complex disease because of what you said. Because in less complex conditions, we're a lot further along in understanding how to use genetics to inform treatment. And I feel like complex disease is that area where it's such a black box, and this is where the work needs to be done. But I definitely think that something like Fragile X, I mean, you're still talking about a patient who's going to have a comorbidity. They may have to be treated with an ACE inhibitor someday for high blood pressure. That's all still relevant. Never is there a patient, I don't think, who has only one disorder and nothing else.
Yeah.
And so, yeah, I think that's a big goal. And I'm hoping to be focusing in on a future grant that really gets at what can we pull from the electronic health record into the pipeline. So that this information could actually go out of being in a mobile app and go into being an alert at some point.
Yeah.
So I know there's a lot to go into that. So
well, I mean, no, I think the general plan you have, it's great. I mean, I really do. We have to do these things. One of the weak areas in it, you sort of talked about at the start, was the fact that the clinicians who are seeing these folks don't have time to pay that much attention to the phenotypes. Basically, they got to keep going, right? There's nobody, right. And so that seems to me to be one of the challenges in terms of really sort of defining the autisms, is the clinicians don't have the time to do it.
Right. No, that's exactly right. And I think so. As I'm a researcher and not a clinician, this is also a little bit selfish for me, because building a pipeline like this, I can sell it to the NIH as a tool for clinical decision support, and that's how I'm going to get a grant funded. But meanwhile, I'm building something that will inform where I spend my money and time. So this is going to pull out some genes, I'm hoping, that are more interesting for me from the perspective of trying to functionally characterize. And so the idea is that hopefully this will be useful to both sides. And right now, really pharmacogenomic variants are probably the most useful information for a clinician, and that's what they're going to be caring the most about. Getting to a drug target that maybe could be repurposed, that's going to be a lot more work for the research community, or to a molecule that's similar to a drug target that maybe we should focus in on trying to identify as a drug target. That's, again, the research community that falls on our shoulders. And so, it's hopefully it's going to be this dynamic process and this an evolution where it's adapting. And so that's why, for example, if you notice in the figure, it's pulling for resources pretty regularly. That's the idea. As the field is changing and advancing and more information gets out there, it's updating constantly. So this information is always new, and like, what's the most cutting edge.
Cool, it's great to have you at KU.
Thank you. I'm excited to be here.
Yeah, but I'll let other people ask you questions and stuff there. There's so many things you could talk about, so. It's good.
Thanks.
Oh, hi, this is Hashim Raza.
Hi.
Hi, so thank you very much for a very nice presentation, and it's a very impressive project, and we really need this kind of, research which can translate to the to clinics. I mean, especially for complex disorders, it's very important. And I'd like to like your idea, using the approach of circadian clock genes, because this is really fascinating, I would say. My question is, since you are focusing on complex disorder and really there are a lot of genes, and that's what we've seen in your second slide, and you're focusing most of those genes which are associated with these disorders based on those common variations, which are in fact also common in the population. So are you also have you also thought about variations in those candidate genes which shown up in the association studies, which are rare in the population? And number two, have you also thought about looking at the genes which are downregulating and also the epigenetic factors in those regions where we see those common variations in the GWAS studies?
Yeah, so that's a really good point. And I would like to note, though, that the resource that I pull the initial list from actually does incorporate if it's been published and reported in the literature, regardless of the level of evidence that's supporting that relationship, that gets put into DisGeneNet. And so that's one nice thing about it, is that what I'm starting with should be most of the information that's out there, regardless of whether it was GWAS or candidate gene or whatever. Now, at the point of trying to prioritize, I do think that both things that you brought up are really good to think about, because right now I'm not really incorporating information about how common or rare the variant that's associated is, and that is something that might be useful. And also whether or not there's evidence for epigenetic factors, which may not actually be as well characterized in the database I'm pulling from. So I think that those are both really important things that could be incorporated. And I would say that one of the future goals of the project is to actually focusing on a gold standard, which may be ClinVar reports or well-known pathogenic variants, and seeing how often I'm capturing those in my prioritization. You could think of a supervised machine learning approach where you're